Okay, hello. This is a video I'll be starting about what I'm learning in my class, physiology class, to mostly help me review because I always find it that teaching helps a lot for me to study. So I'll be teaching what I've learned. Hopefully I didn't learn it wrong. So this is the first week of my physiology class. Now let's begin. The first thing we talked about is homeostasis. Just learning about homeostasis because homeostasis is how the body is not dead. So homeostasis, it's described as dynamic constancy. So dynamic constancy means that it's staying constant, but it's always changing. So how does something stay constant and is always changing? Well, homeostasis is the dynamic constancy of variables within a set point. So the example we use in class to have a set point, so there's two set points. Let's say we use temperature. So there's two set points, and the constancy is staying within the set points. But being dynamic is because it's always changing between these two temperatures. That's homeostasis. Um, the way we maintain homeostasis is sometimes organisms, they exchange matter and energy with the environment. Or they also exchange, or organisms receive signals from environments and respond. An example we use in class is for the second one. If you're cold, of course you'll shiver. That is your body telling you that you have to maintain your temperature. And then if you shiver, or maybe uh, I guess the first step will be rubbing yourself to keep warm, then shivering so that you can keep even more warm. And once your body has reached the temperature that you'll feel comfortable at, then you've achieved homeostasis, and your temperature is changing to reach that state of constancy at the temperature that you should be at. So we learned about Claude Bernard. He studied homeostasis, and he said that there's a constancy of the milieu interior, which means that the cells are constant in the environment that they have. Now, a guy named Walter Cannon, he talked about homeostasis, the maintenance of it, and it's maintained by this is the homeostasis. So it's maintained by negative feedback loops. Positive feedback loops. Just a bunch of loops. Um, Competition of loops. And hierarchy of loops. Oops. Cover. Redundancy of control. I don't know what that means. And I don't know what the last one means either. It means steady rate of parameters that requires energy. Okay, whatever. Don't want to write that out. So that's what Walter Cannon talked about maintenance. Then we talked about cell physiology. We first started off with the cell membrane. So cell membrane is also called the plasma membrane, and it's made of a phospholipid bilayer. That means, well, phospholipid is just fatty acids. These are chains of hydrocarbons. They're attached to this 3-carbon thing. And then over here there is a phosphate group, like that. And this makes it polar, so the phosphate group will be hydrophilic because it's polar. It will dissolve in water, or it likes water. It'll be attracted to water since water is polar also. And these fatty acids will be hydrophobic, so it doesn't like water because it's non-polar. It's just a bunch of hydrocarbons, which means it's just hydrogen and carbon. Hydrogen and carbon. So bilayer means that just 2 of them makes a whole layer around the cell like that. And that's a cell. And that's what they call the head. This is the head. And these 2 are the tails. Okay. Also, this means that since it has a hydrophobic interior or in the middle, that means that the plasma membrane is selectively permeable. So that means not everything can go through it, which is good. There's also 2 different types of transport. There's active and passive. For active, there is primary, which means it uses ATP as energy, and then there's secondary, so it uses other molecules as energy sources, which can be ions and other stuff that I don't know. Now, passive transport means that it'll go, there will be a gradient, and it will go, the stuff will go from a higher concentration to a lower concentration. Whatever it is, whatever solute it is. Now, let's go on to the nucleus. Nucleus contains DNA, of course. DNA is in there as chromatin. These are just this is the structure of DNA when it's all crumbled up, bunched up together because of hydrogen bonds and stuff. And there's a nucleolus. Inside the nucleolus is where ribosomes are made, or RNA, to make ribosomes. And the nucleus also has a phospholipid bilayer. And in the phospholipid bilayer, there are what are called nuclear pores to allow small molecules and ions inside the nucleus. Now let's talk about cytosome, or cytoplasm. So the cytoplasm contains cytosol, contains the cytosol. So cytoplasm just means it's whatever's inside the cell, everything that I've darkened, which means it's everything. That's the cytoplasm. Cytosol is the liquid matrix inside the cell. So here's the nucleus. Nucleus is part of the cytoplasm. Cytosol is the liquid matrix that suspends everything inside the cell. There's also, of course, cytoskeleton to keep the cell structure. And there are also, of course, organelles. These organelles, of course, they'll be like around the cell. These are all part of the cytoplasm. So regarding the cytoskeleton, the cytoskeleton is a scaffolding of proteins. And this means that there are a lot of proteins in the cytoskeleton to keep the cell structure. Now they are filaments. First one is microfilament. And microfilaments are categorized into two. There's a thin microfilament, which is 5 to 8 nanometers wide. There's thick microfilaments, which are 10 nanometers wide. So the thin one are called actin. Thick one are myosin. Actin is dynamic because it's called dynamic because here's an actin. This one, there's always one end that's constantly growing, one end that's constantly shrinking. That's why it's called an active or dynamic. That's why it's called dynamic. Now there are also intermediate filaments. And these are about 8 to 10 nanometers wide. An example are neurofilaments. And then there are microtubules. Microtubules are pretty important. They're 22 to 25 nanometers long, or wide. They're pretty important because you can find them in flagella, which is the tail thing or whip that cells use for movement, or bacteria. Cilia is also for movement. It's also found in the spindle fibers of mitosis and meiosis. Remember, spindle fibers, they're the things that come out of the centrioles to grab the middle of the center, the chromosome, to split them apart. They're also organized in the centrosome. Sorry, my handwriting is really bad. And the centrosome organize it. They also transport vesicles. Another role that they have is to anchor membranous organelles. So any organelles, here's a cell, any organelles here and such are anchored to the membrane by these things, which are microtubules. I think that's it. Okay, the next thing are proteins. So proteins are really important. So proteins have many destinations. Of course, we know proteins are made in the ribosome, and if you don't recall. So DNA to RNA to protein. Whatever. So DNA to RNA, that's called transcription. And RNA to protein is translation. Okay, so proteins are translated in the ribosome. Our ribosomes are made from rRNA that are made in the nucleolus. And there are many destinations for proteins. So proteins can go to the cytoplasm, where they can finish their translation in free ribosomes. I won't write that. Next, it can go to a cell or organellar membrane. So those proteins will probably be like channel proteins to allow molecules to go through them, like a gateway. Or it can be used inside the cytoplasm as transport for other proteins. They can be transporters, like a bus, for other proteins. Proteins that will get on that protein bus. That's a protein. Or, so we'll go to membranes. Could be used to make organelles and such. Or it could be secreted out of the cell, deposited into the lumen of the endoplasmic reticulum vesicle. So what else about proteins? You learned that in the rough endoplasmic reticulum. Okay, so you know that proteins are, let's say this is the nucleus, this is the cell. There's a rough endoplasmic reticulum here. And then there's smooth endoplasmic reticulum. Smooth endoplasmic reticulum is kind of a tube-like thing. And they're connected. And here's the Golgi apparatus. Okay, let's say that there is an RNA sequence that comes from the nucleus that's translated there, and it's meant to go to the rough endoplasmic reticulum, or they're actually destined for secretion. Proteins destined for secretion. So we'll go over that. So proteins destined for secretion will go into a vesicle, and the vesicle will spit it out. Alright, so the first thing, here's a protein. It's not finished yet. And the protein will come out of the nucleus and it'll wait there. Now on the protein there will be a 15 amino acid sequence. And this is a recognition sequence, so that it will be grabbed by a protein that is sent out by the rough endoplasmic reticulum, something here. And that protein has a recognition sequence on it to be able to mount on a receptor on the rough endoplasmic reticulum. So let's say it looks like this. Yeah, something like that. So it will detect the 15 amino acid sequence right here on this RNA, and it will grab it if it detects the right 15 amino acid sequence, and it will carry it, it will transport it to here. This protein will go attach here. This protein will go inside the rough endoplasmic reticulum. And so it will continue to be translated inside the rough endoplasmic reticulum while it moves through it over here like that. And this is called co-translational translocation. So this means co-translational, that means while translating at the same time while it's moving, translocation, changing location. So that's what it does in the rough endoplasmic reticulum, and of course there are ribosomes in there to do the translation. And so the protein will move to the smooth endoplasmic reticulum, and then they'll move from the rough, smooth endoplasmic reticulum into a transport vesicle, right here, transport vesicle, which will move it to the Golgi body, where post-translational processing can happen. Now, while it's in the rough endoplasmic reticulum, there can also be post-translational processing, where the 15 amino acid sequence can be deleted there. Also, you can add a sugar to the protein, and this is called glycosylation. It will make the protein into a glycoprotein, which just means sugar plus protein. Sugar was attached to a protein. It can also have disulfide bond formation, which contributes to protein structure, tertiary protein structure, and folding, and also hydrosylation. These are all post-transcriptional processes. Modification of proline residues. Okay, that's what happens in the rough endoplasmic reticulum for post-translational stuff processing. Now when it moves after the smooth endoplasmic reticulum into a transport vesicle to go to the Golgi body, the Golgi body will also do some processing post-translation. It can also do glycosylation, Golgi body. It can also cleave some sugars it doesn't need, and it will sort proteins to their final destination. So once the protein that is destined for secretion is sorted in the Golgi body, let's say it's over here, usually it's sorted out in the farther bottom parts. Then we move on to secretory vesicle formation. So imagine this whole thing's a cell. This is the Golgi body. Protein moves out of the cell to the plasma membrane. Goes to the plasma membrane. Wait, I'm just kidding. It does not go to the plasma membrane just yet. The protein is inside the Golgi body. Well, there are probably many proteins here. And this drawing will probably be very wrong. But it's not meant to be truly correct. It's meant to review. So in the cytoplasm, there are these things called cotomer proteins. Okay, so the first thing that will happen is there is a thing here. Something, I don't know if that's called, a receptor, where the protein that we made will attach to it. And once the protein, let's say, attaches to the receptor, then that will make the cotomer protein also attach here. And then let's say that happens a couple of times. Happens a couple of times. Cotomer proteins. I don't really remember how they look. Let's just say I know they look like that. Such. They coat. And when there's just enough cotomer proteins, what will happen is that this part will branch off. It will make a bubble around the proteins. So now it's cut off. Say it's cut off. And now these proteins so now there's all these cotomer proteins around. And it goes into the cytoplasm like that. Now we have a vesicle. Now what happens in cytoplasm is that the cotomer proteins will then dissociate. So then there's no more cotomer proteins. And in the vesicle there are these things called V snares. It's a very bad drawing. So I don't have another marker. So let's say there are these V snares right here. These are V snares that were under the cotomer proteins that are now revealed once the cotomer proteins dissociate in the cytoplasm after this whole thing becomes a vesicle. Now what happens is that the vesicle will travel to the plasma membrane. Let's say this is the plasma membrane. Vesicle will travel here, and in the plasma membrane there is a receptor for the V. This is called a V snare. At the plasma membrane there's something called a T snare, and the vesicle will come and attach over here. Whoop, like that. And eventually the vesicle will wait there for a signal for exocytosis. That means it will release its contents to outside of the cell. So the result will be like this. Bleh. Exocytosis. Bleh. Everything will go out. And that is the point of proteins that are destined for secretion. That's exocytosis. Now we also learned about endocytosis, which is stuff being absorbed by cells. Now there are the many reasons for endocytosis is because, of course, when you do exocytosis like this, let's say cells like this, a vesicle attaches, it'll open up the cell from here and actually extend. This is longer now. It will extend or lengthen the plasma membrane. However, there is also endocytosis where the cell will take in new stuff from the outside. Reasons for it are: one, it will take in stuff, movement of nutrients. So cell or nutrients that are too large to be imported by transmembrane proteins. So that means there are some nutrients that are larger than the size of the pores of the channel proteins, and they can't get in. So the cell will do endocytosis to absorb these nutrients. Second reason could be to stop signal process of hormone receptor complexes. I don't know what that is. I assume it's for the cell to take in stuff that maybe activate hormone secretors. Yeah, so that the stuff will be dissolved in stuff in the cell. What are they? Lysosomes? Something that destroys stuff. Anyways, the third one is to shorten or degrade part of the plasma membrane. Terrible handwriting. Alright, whatever. So there are, how many kinds? There are 3 kinds of endocytosis. The first kind is called pinocytosis. Now, pinocytosis, one good way to imagine it is if you're underwater and you breathe in, whatever you get is whatever you get. You don't get to choose what you get. Same thing with a cell. So whatever's outside the cell, here's a cell. Whatever's outside here, whether it be poop or water, it's going to take that in. The cell will be a bit smaller. Now, one thing that happens in endocytosis is that inside the cell, after whatever, or inside the cell, the codimers, they're being shown again. Codimers are pretty important for the cell to make a pocket. As it seems, codimers will come from inside the cell instead of outside, and it will attach to the plasma membrane. So the second, oops, dropped my marker. The second type of endocytosis, oh by the way, this is called fluid phase endocytosis. Second one is called receptor mediated. So that means it can choose what molecules the cell wants to absorb. So there's a target molecule that has a sequence that will match with a specific receptor. And once that molecule is attached to that receptor, then of course more codimers will come from the inside and make a pocket, make a vesicle to absorb whatever molecule is chosen. And the last type, it's like eating, phagocytosis. So an example, it's just an attack. Here, a white blood cell. And so this is what are called pseudopods. White blood cell attacks something, let's say this is a bacteria, will attack, and then it will eventually absorb it. So it goes like this. That's phagocytosis. And then the last thing we learned today is apoptosis, which is programmed cell death. So apoptosis is tidy, clean. Wow. I'm looking through my notebook and there's quite a bit that we learned. Whatever. Tidy, clean, clean death. And there's also necrosis, where it's a messy death. And it's usually destruction of damaged cells. Those are the two types of cell deaths. Now, we move on to next chapter, which is plasma membrane and membrane potential. Okay, so we learned that all about cholesterol. I know that in the plasma membrane, cholesterol will come in here and help make the membrane more fluid or flexible so that the membrane can bend and twist and such. Because if cholesterol is not there, then the lipids will crystallize. So cholesterol prevents fatty acid crystallization. So cholesterol is somewhat important, but you don't want too much of it. And what happens when fatty acids crystallize, it'll make it more stiff. Not good. Another thing that I learned is that the phospholipid bilayer can move because it's fluid. Because of the fluidity, it can move. Cholesterol can also flip. Can flip, I don't know what that means. Okay, so there we go back to proteins. So proteins there are integral proteins, and integral proteins are embedded in the plasma membrane. So here's the plasma membrane and stuff. Integral proteins are like channel proteins, like that. And there are peripheral, which are proteins that are just in the cells like this, like that. Those are peripheral proteins. They're anchored by weak bonds. Now, we talk about the extracellular matrix. I'm gonna miss some stuff. Like we talked about ligand receptors, also adhesion molecules. We also talked about lipid rafts. They're composed of sphingolipids, which I assume they're not fatty. They are not phospholipids. They have this thing called sphingol instead of phosphate. And they have extra cholesterol. I guess that's cool. And these lipid rafts are tightly packed. And highly organized. There are also lipid flip bases. You don't know 100% what the functions are, but possible functions from research. Possible. Or it can be used as transport, or it can create or maintain essential bilayer. So there's that. Alright, so the extracellular matrix. It is what is outside of the cell. So here's the cell. Extracellular matrix is what's outside. The surroundings of the cell. And it's made up of 3 protein fibers. So the extracellular matrix is made of collagen, elastin, and fibronectin. So cells, of course, like my hand, which is made up of a bunch of cells, they need to stay together. So we talked about cell to cell adhesion. So the first cell to cell adhesion is called a desmosome. 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 And so there are 2 cells, and they're made up of this thing called cadherins. And the cadherins are attached to these areas of thickened They're thickened cytoplasm or cytosol called plaque. And the plaque has these things sticking out of it, and this is keratin. There's something else called so the cells, they don't touch, as you can see. There's distance between them. There's also a desmosome, where a cell is attached, the cadherin is attached to the extracellular matrix, and it will be attached to specifically fibronectin. Cadherin will be attached to fibronectin. So that's the desmosome. Okay. It's the strongest, by the way. I didn't mention that. Desmosome is the strongest adhesion. Alright. So the second cell adhesion is called a tight junction. Tight junction. So here's a cell. So here are the plasma membranes. And at the plasma membranes, there are proteins right here. And these proteins are called Claudin proteins. This area is called a kiss site. So the Claudin proteins on the kiss sites, and these tight junctions are at where cells need to not allow stuff to go through, so they're tight. Not allow any penetration. So it's found in digestive tract, because you don't want any of that nasty stuff to get out of the digestive tract. So there could be many layers of this between two cells. The last one is called a gap junction. So there is, of course, a gap, and there are two cells, and the point of the gap junction is for communication. Allows import and export. So allows molecules to go through. So in between two cells, there's a passage of right here. There's a passageway between them. And these are composed of connexons. And it will allow only small molecules and ions to pass through them between the cells. The distance here is about 2 to 4 nanometers. The last thing we talked about is membrane transport. So we already know passive, which is because of a gradient. We're talking about diffusion. Diffusion is in regards to solutes. There's a equation by Fick's law of diffusion. So Fick's law of diffusion, it's Q equals change of C times A times P. Q is net rate of diffusion. And the problem with this is only takes account of concentration gradients. And we know that ions also pass through, which makes an electrical gradient. No electrical. That's the problem with this equation. Then we learned about osmosis. So osmosis is the movement of water, or diffusion of water. So there'll be a higher concentration of water on one side. Say this has a higher concentration compared to this. Water will want to go this way to the lower concentration. So type of passive transport. However, when something's at an equilibrium, let's say here's a membrane, make it smaller. So here's an example. The membrane in between here, and then there's a membrane in between here. So let's say we have this much water, no solutes, and we have this much water with some solutes. Membrane is only passable by water, and so the water will of course move because of osmosis this way, because of diffusion, of course, because diffusion happens when there's a gradient. There's a gradient of water, more concentration here compared to here, will move this way. So let's say the water level becomes like this, and then this water level becomes like that. Now the water levels are not the same, but why is it at equilibrium like this? That's because of the solutes here. There is an osmotic pressure. So there is movement of water from here to here due to osmosis. That's due to the gradient. And there's also an equal amount of movement from here to here of water due to osmotic pressure. Now, We also learned about tonicity. And I'll go over this later because I don't really understand it too well. And that's it. Finally, that was so long.